running a little behind and frazzled this week for whatever reason, so I don't have the slides posted for today, but I'll try and get them up later on today. I was also unable to find my old copy of the homework problems for working with single gene, single trait crosses, and then with dihybrid crosses, and also dealing with pedigrees. So I started Monday afternoon recreating that thing from scratch, and I don't know, I guess I'm about halfway through. It's going to cover stuff that we're going to be covering on Friday as well as Monday and Wednesday, so I will try and get it out to you by the end of the week. Maybe tomorrow, just depends on kind of how the rest of today goes. Because it'll help you to get practice doing that in preparation for having to do that on the test. So from last time, do you guys all feel confident in how single trait, single gene crosses work? How do you feel about applying probabilities to those crosses? Do you feel okay? You look less convinced in your abilities there. Could you do a chi-square test if I gave you an outcome of a cross and asked you to compare it to an expected phenotypic ratio?
No.
Yes.
Yes. You couldn't do it well.
It is just an equation.
Yeah.
Jamie, what's your difficulty with it, do you think?
Need to see it up live, I think, before it would be like super sketchy, but.
Okay, all right. Do you want to go through an example now, or?
Sure, yeah.
Okay. Okay, so you can go online and find Mendel's results. You don't have to go and read his original paper or anything. But basically, these are some single gene, single trait crosses, and it basically tells you what he found in the F1. Why do you want to know what's in the F1? What the phenotype of the F1 is? Because that's the thing that's going to be dominant. So anytime you take two different traits that are true breeding, so you have true breeding round and true breeding wrinkled, the phenotype that comes out in the F1, that's the dominant, so now you know how to orient your expectation for what's in the F2. And what you should expect in the F2 is you should expect more of the dominant phenotype than the recessive phenotype. So we dealt with this one the other day. We could look at yellow versus green seeds. Yellow seeds in the F2, he had 6,022. He had 2001 green. Clearly, because of the F1, yellow is the dominant phenotype, green is the recessive phenotype. We would assume that the phenotypic ratio of the F2 would be a 3-to-1 ratio. And so if we have 3, 2, 0, 8, we have 8,023 total seeds that were scored for color. What would be the expected number of seeds that would be yellow if it was a 3-to-1 phenotypic ratio? figure that out?
8,023
Not divided by 3. If there's 3 parts that are yellow and 1 part that is green, that's 4 parts total, so it would be 3/4 of that and 1/4 of that. So we would multiply 3/4 times this, right? So 8023 times 3 divided by 4. What would that be? Somebody with a calculator. Jimmy's gonna do it. Does he have a question?
6,017.25.
And what's 1/4 of 8023?
2005.75.
2000 what?
5.
Yeah, you could also do it by dividing it by 4, finding that number, and then subtracting that number from 2023. This should equal 8023. Let's check it and make sure that it does. 13 to 2023. Okay. So one of the confusing things about chi-square tests is there are a bunch of different kinds of chi-square tests. They all rely on the same equation, but how you set up the test is different. So this is what's called a goodness of fit test, where you're basically taking some raw data and you want to know how well those data fit some sort of expectation. When you do a normality test, one of the ways that you can test for normality in data is with a chi-square test. It's not the best way to do it, it's just one way you can do it. And you're basically testing to see if the data that you observe conform to the expectation of normality determined by a theoretical normal curve. So in this case, we're basically trying to figure out is if this distribution of yellow to green seeds conforms with some expected distribution based on, in our case, our knowledge of Mendelian genetics. And so it's what's referred to as a goodness of fit. And when you do goodness of fit tests, you actually generate the expected values not based on the data themselves, but based on what your theoretical expectation is. And so in this case, the chi-square test is made up of the summation across all the categories, and we're going to have 2 categories here, of the observed minus the expected. And what do we do to it? We square it, and then we divide it by the expected. So in this case, we would have 6,022 minus 6,017.25. We would then square that, divide that by 6,017.25, add that to 2001 minus 2005.75, square that, divide that by 2005.75. And somebody with a calculator, what does that equal? It's going to be really, really small.
And you want that answer to be less than 0.05?
No, this is the chi-square statistic. So I'll pull up a chi-square table in a minute.
Okay.
So this is the actual chi-square statistic that you're going to evaluate at 1 degree of freedom.
0.0149.
0.0149 is the total here.
That's what it says. It's good in the books.
This is tiny. This is a teensy-weensy chi-square statistic because there aren't a lot of deviations. It's 6,022 versus 6,017. That's not a lot of difference between the observed and the expected. So on the test, I'll give you a chi-square table. So So if you're testing things at a 5% type 1 error rate, we have 1 degree of freedom because we have 2 categories. We would have to have a chi-square statistic of 3.841 or larger. To say that the observed distribution is different from the expected distribution, and we don't have anywhere near that. And so that's a 95% confidence or a 5% type 1 error rate. Remember, type 1 error is the odds of saying that 2 distributions are different when in actuality they're not. We're nowhere close to being able to say that these 2 things are different because our observed chi-square value is 0.0149. And you have to be bigger than that. Remember that as your expected differs from your observed, this gets bigger. The more categories you have, the whole thing gets bigger. And so you see that as you increase the number of degrees of freedom, the criterion for rejection gets bigger and bigger because you're adding together more and more categories. And so you have to have a bigger chi-square test. So when we do these tests for a dihybrid cross, where your expected phenotypic ratio will have 4 different categories for homozygous dominance for both traits, homozygous dominant for the first, homozygous recessive for this, this is basically dominant for both traits. This is dominant for the first trait, recessive for the second trait, recessive for the first, dominant for the second, and recessive for both traits. Those phenotypes, you now have 4 possibilities. So you would use 3 degrees of freedom, which you would need to have a larger chi-square value to get to significance if there were differences. Not because of anything else other than the fact that you have 4 categories that you're comparing now rather than 3. And so,
So if it's less than that value, it's not significant,
if
it's
it's-
not different.
yep, if it's less than this value, then it's not significant and it's not different. Because this difference is what's driving the whole thing. And so as differences between observed and expected get bigger, the number gets bigger, and the bigger those differences are, the more likely you are to say that they're different. So you're looking for big numbers of the chi-square test. Big numbers of the chi-square test would lead to small numbers of p. So p less than 0.05. And in this case, for this number, p is much bigger than 0.05. Well, we can go online once again like we did last time. Oh, I've never seen Daniel Soper's face until just now. What was it, 0.0149? 1 degree of freedom. Anybody want to guess what the p-value is going to be? 0.2. Oh, it's going to be bigger than 0.2. I would guess it's going to be closer to 1 than it is to anything else.
So, at least
0.90. So you could say that this distribution is different from this distribution, but you would say that and be wrong 90% of the time, essentially, is one way of thinking of what a p-value does. So this is a gargantuan. P-values go from 0 to 1, and this is much closer to 1 than it is to 0. And I could pull up any of Mendel's data and make you do this with them. This is why sometimes people think Mendel cooked his books. Because really, really tiny chi-square values when you run a chi-square test on it, and really, really huge lack of significant differences between observed versus expected. Does that help at all, Jimmy?
Yeah.
So remember, it's always going to be observed, which is what he found, versus expected, which is what you would expect, given a particular form of inheritance. Now, on Friday, we're going to talk about some phenotypic ratios, and today we're going to talk about some of them that turn out to not be what you would expect using normal Mendelian genetics, because there are phenotypic ratios that arise because genes are found on the same chromosome, so they're genetically linked to one another, so that they get inherited together. In the case of genes that are dose-dependent, such that the number of copies of a gene determines phenotype, you would expect the phenotypes to be in a 1 to 2 to 1 ratio, because the homozygous dominant gets 2 doses of a gene, the heterozygote gets 1 dose of the gene, and the homozygous recessive gets no doses of a gene. So in this case, you wouldn't, if you were expecting a 3 to 1 ratio, the first thing that you would find is that you actually have 3 different phenotypes, and those phenotypes would be distributed the same way the genotypic ratio is, because these are genes whose phenotype is dose dependent. And so we'll see, but then you could test your observation of offspring against a 1 to 2 to 1 ratio and see if it differed or not. All right, other questions?
Ask about the encoding bugs. Like, why do people think that? What if it's just something that works really well for
Is-
a single loop?
Well, his data are just really clean, and that's why people think it. But once again, the argument could be said that he was scoring thousands and thousands of seeds, and if you score enough seeds, then you eventually converge on what the true answer is. So, like I said, I'm not terribly interested in that question, because it turns out that Mendelian genetics works. So, yeah, well, I would not want to sit around and count 8,000 plus seeds to get this kind of an answer. Clearly, he was more devoted than I would be to this question. Yeah. Katie?
You said the p-value was the probability that an event will occur, or?
So it's the observed type 1 error rate. So there are 2 ways in which you can make an error in statistics. One is that you conclude that 2 things are different when they are not. And so that doesn't that applies to like comparing 2 means. If your null hypothesis is that they're the same, and you conclude after doing a t-test on them, for example, that they are not the same, that maybe one is bigger than the other, you can conclude that, while actually the truth being that they're actually not different. But it could also be testing 2 distributions to see if they're different. Distribution A versus distribution B. These distributions could be different because one is a unimodal and the other one is bimodal. They could also be different because one of them is further down on the numerical X scale and one of them is further up. So it doesn't matter whether you're talking about differences between distributions or differences between means. Just you're saying 2 things are different when in actuality they're not. The type 2 error is concluding that 2 things or more things, and it only has to be at least 2, it could be multiple things. The 2 things are not different when they are different. So you would analyze these 2 distributions and you would say that they're not significantly different when they actually are. And so we normally have this thing called alpha, which is the type 1 error rate. And we set this before we ever do an experiment. And the type 1 error rate is basically just how often do you want to make this kind of an error? Do you want to make it 5% of the time? Do you want to make it 10% of the time? Do you want to make it 1% of the time? You, the researcher, get to set what this is. The standard in biology is that this is 0.05 because that's a 5% type 1 error rate. You don't want to say two things are different and have them actually be not different, only about 5% of the time. But that varies somewhat. So when I worked in Puerto Rico, we did this huge experiment. It took us 18 months to set it up, 18 months of having full-time crews in the field setting it up, because we hired these arborists to go into the forest and they cut the canopy of the trees out because we were studying hurricane disturbance. And so rather than wait around for a hurricane, we simulated hurricane damage. And so we had 12 different plots around the forest, and in one of those plots we left it alone. In another plot we went in and we cut the canopy out and dropped it on the floor of the forest, which is exactly what a hurricane does. It knocks down a bunch of branches, dumps them on the forest floor. But you're changing two things when you do that. You're changing climate because there's no longer a canopy buffering climate, and you also are changing how much organic matter is decomposing and feeding the community of organisms that are in the forest. And then in another set of plots, we hired arborists to go in and cut the canopy out. But as the canopy came down, we had a crew of people on the ground who weighed the canopy so we would know how much was coming out of the canopy. And then we picked it up and moved it to a closed canopy place and dropped it on the ground over there. So now we have a closed canopy place where there's litter addition, so we can look at the effect of just putting litter on the ground, increasing the amount of nutrients that are there. And then we had a place that we had opened up the canopy, changing the climate, but there was no litter there feeding the community, so we could decouple these two things. It costs a lot of money and a lot of time, so we only had 3 different replicates. We replicated these, we had 4 plots in a block, a control, a cut and drop plot, a cut only plot, and a drop only plot. And then we had 3 of those. So we basically had a sample size of 3. And so when we were setting this up, we asked, is a sample size of 3 enough to detect a difference if the difference is actually there? And what we decided was the size of the disruption that we were causing to the forest was going to be huge. And we also discussed that that would probably give us enough power, enough statistical power to detect differences if they occurred. But one of the other things we decided is, do we want to stick with this, or do we want to relax our alpha level so that if a difference does occur, we can catch it? And the nice thing about being able to do this is you can say, well, what's the consequence of saying there's an effect when there isn't? Well, there really isn't a consequence. Nobody's going to die because a group of ecologists working in a tropical forest in Puerto Rico decided that decoupling climate from nutrients actually had an effect. I might offer, who cares anyway? Ecologists care. Ecologists care passionately sometimes. But in the grand scheme of things, getting the wrong answer, making a type 1 error, the world's not going to end, nobody's going to die. And so we also discussed whether or not we just, for everybody who worked on this project, and there were 23 different researchers working on this project, did we all just want to adopt a relaxed alpha level? And we sometimes see this in literature. Now, take for example a health care issue where maybe you have a cure for glioblastoma. Glioblastoma is a perfect type of brain cancer. Basically, if you're diagnosed with it, you're doomed to die in at most a couple of years, usually in about 6 months. So it would be really nice if somebody could find a cure to glioblastoma. And so somebody thinks that they found a cure to glioblastoma, and they're pretty sure that it works, and they get it to the point that it's doing human trials, and you give it to a bunch of people, and indeed, it cured the glioblastoma. It eradicated the glioblastoma, but it was so toxic to the body as a type of chemotherapy that it also fries your liver. So now you're cancer-free, but you need a liver transplant. Do you want to say that the control group that got a placebo versus the drug group are different and risk making a type 1 error saying they're different when they're not 5% of the time? How comfortable do you feel about that? It works, but you need a new liver. Do you want to make a type 1 error 5% of the time? Catherine, how often do you want to make a type 1 error?
I don't know, 1%?
Okay, so in this case, you would maybe make your type 1 error rate more stringent. And actually, you see this a lot in medical research. You see this a lot in medical research because there's this balance between, do you want to bring a new therapy onto market? If you do, you want to make sure that it works because almost everything that we do to our bodies has some sort of side effect. And so you have to weigh, is the thing that you're advocating actually effective versus what the downsides are of the side effects? The worse the side effects are, the more sure you want to be that the thing is actually doing what it says it does. And so you don't want to necessarily make a type 1 error 5% of the time. Now, what's the problem with reducing the amount of times, the probability of making a type 1 error? When you reduce the amount of, when you reduce the probability of concluding that two things are different when they're not, to 1% or maybe 1/10 of 1%, what do you end up doing?
Increasing the likelihood for type 2.
You're increasing your type 2 error rate. Because something that is actually working, you're concluding that it doesn't. So the faster you run away from type 1 error, you are running into the waiting arms of type 2 error. Most biologists don't think about type 2 error. But they should, because as you try and avoid one type of error, you run headlong into the arms of the other type of error. And both of them are kind of equally bad. In one case, you're saying things are different when they're not. You're seeing a difference that doesn't exist. In the other case, you're not seeing a difference that does exist. And so you've wasted all of your time doing this experiment only to ignore a difference that is there that you're not going to acknowledge. And so you have to value, you have to have an internal discussion when you do research. Which of these two types of error do I value most? If you read papers where all they talk about is something significantly different or not, we value this type of error more. We think that's the more important type of error to avoid. I would argue, and I argue this when I teach applied statistics, that that's not necessarily what we should be doing. We should be paying attention simultaneously to both, but it's harder to think about both at the same time. So this is what, so P then, P is the observed type 1 error rate. And so is your type 1 error rate bigger than or smaller than what you wanted to see when you walked into the experiment? What you should never do, though, is go and gather your data and then decide what your p-value should be. So you always want to decide on what alpha should be beforehand. Otherwise, you cook the books in another way. You just pick the p-value that shows that something is different, which is not really good scientific protocol at all. Okay? Does that help?
Yeah.
Okay. So probably more than you were asking for.
No,
but
it was great.
okay. All right. Ask me how to drive from here to Columbia, and I'll tell you how to build the car to do it. Okay, other questions? All right, let's talk about different kinds of inheritance. So as you look around the room, you will see that there are people who are different sexes. We have males and females, and these things are determined, in some cases they're determined genetically, and in some cases they're determined environmentally. And so when we think about inheritance and sex, there are these sex chromosomes that some organisms have. Humans have them, X and Y. Birds have them, they're Z and W. They work slightly different in terms of sex determination. But Insects have them, but it's just a matter of the number of X chromosomes that you have, not that you have one of one type and one of the other type. So there are different ways in which sex chromosomes go about determining sex, but because you have these different sex chromosomes, there are genes found on the Y chromosome that are not found on the X chromosome, and vice versa. And so you get interesting and different inheritance patterns when you think about the genes that are on those special non-autosomal chromosomes. And so they lead to unexpected phenotypic ratios when you do kind of standard crosses. And so one of the goals of this chapter is to get you to explain how those things work. And we're going to look at a lot of different crosses so you can see how that's working. Also, there are differences in sex determination depending on what group of organisms you have. Placental mammals do things one way, birds do things another way, insects do things a third way. And so there are lots of different ways of determining sex. Reptiles do it a totally different way in some cases. And then one of the special cases of this is temperature-dependent sex determination, which is commonplace in reptiles, although it's not found in other organisms. And it's interesting kind of how that works. So we'll go into that in a little detail, a little more detail than what is in your book, but it actually fleshes some things out that your book doesn't flesh out. The book left me a little hanging on this point. So. We are a species that exhibits sexual dimorphism, which are these physical differences, phenotypic differences between the two sexes. In many cases, sex is determined by the presence of sex chromosomes, either XY or ZW. And the genes on these X chromosomes are known as X-linked genes, and they have special patterns of inheritance. You cross two individuals, and because there are genes on the X chromosome, they get inherited differently because in a male, in humans, there's a gene on the X chromosome and there's no equivalent of that gene on a Y chromosome. And so it's as if the chromosome isn't even there. And like I said, there are lots of ways of thinking about this. So in our karyotype, so this is just a human karyotype, an actual imaging of the chromosomes. Females have two X chromosomes. They're both nice long chromosomes, and in males we have a nice full X chromosome and we have a dinky little Y chromosome. And so in all of these cases where you have chromosomally based sex determination, there is always going to be one chromosome that is really big and one chromosome that is really tiny. And this is the one that is usually referred to as the different one, essentially. So this is the X and this is the Y. Not only does it have fewer genes because it's a shorter chromosome, in our case, it also has different genes on it. And so that becomes important because you can have X-linked genes, but you can also have Y-linked genes, genes that are only found on the Y chromosome and are not found on the X chromosome. So if we go back to fruit flies, and your book does a pretty good job of relating this story, the first mutation that was found when Thomas Hunt Morgan, which is a name that you should know, he's the guy that kicked off the whole Drosophila thing in genetics. He was a researcher at Columbia University back at the turn of the last century, the early 1900s. And between 1900 and about 1910, he discovered most of the major things about inheritance and linkage and things like that that could be known, essentially. So he had a really productive first decade of the first century. They rediscovered, 3 different labs rediscovered Mendel's writings from the 1860s in the year 1900, and as soon as that was known, genetics kind of really took off at the beginning of the 1900s. Thomas Hunt Morgan created at Columbia this lab. He had a thing called the fly room where he was raising just tens of thousands of fruit flies. The nice thing about raising so many fruit flies is you now have enough individuals that rare instances, like mutations, began to crop up. And so if you raise enough flies, you're eventually going to see some mutations popping up, simply because mutations happen at a particular rate. If you have a mutation rate you observe a mutation about 1 in a million times. If you have 10,000 genes, you only need 1,000 flies to start seeing, is that right? 1,000 flies to start seeing mutations arising. And one of the first mutations that he observed was this white eye mutation. It was the first one that he observed. So normally, fruit flies have these nice red eyes. That's the wild type. And so in fruit flies, we put pluses on wild type. And presumably he had lines and lines and lines of true breeding flies, because every parental cross always produced red flies. And then all of a sudden, he observed a mutant. And your book actually doesn't put the minuses. It just has them lowercase without the plus. So the pluses mean wild type, and mutant is not wild type. And when he crossed these with one another, he basically got a bunch of offspring that were all red-eyed offspring. Whenever you take a red-eyed fly and cross it with a white-eyed fly, and you get in the F1 generation red-eyed flies, what does that tell you about red eyes?
It's the dominant trait.
It's the dominant trait. So in this case, red is dominant. When we talk about wild type, what does wild type mean relative to dominance? This is from a couple of chapters ago. What is wild type when we say something is a wild type phenotype? Does it have anything to do with dominance versus recessiveness? No. What does it just mean?
Unmutated. Like unmutated. Natural.
It's the thing that naturally occurs. So it's the thing that you would find if you went out and sampled natural populations. Now, in a lot of cases, wild type ends up being dominant, but it doesn't have to be the case. It's just what's found in normal flies out in nature. This is the mutant because it's the rare one, but there's nothing that said that it had to be recessive to the dominant phenotype. In this case, of course, what are these genes coding for?
Pigments.
Pigments, and so this is a gene that is functional and is producing the pigment, and this is what we would call what kind of mutation? It might be a deletion, or it could be something else that is not a deletion, but a deletion or insertion would be the easiest. But in terms of kind of functionally, what would we call it? Mention this just in passing. The week before spring break.
Is it just not functional?
Yeah, it's what we call a loss of function mutant. And so you've lost function. If you have a deletion, like Mason said, you would actually then disrupt the structure of the open reading frame, and you might produce a protein, but it would be a protein that wouldn't function in the same way. And so you've lost function, you don't produce the pigment that is necessary, which is why you go from having pigment to what would be white ID, not having pigment. And so when he crossed a white-eyed male with a red-eyed female, he got a bunch of offspring that were all red-eyed. Now, ignore for the moment that we are talking about something that occurs on the X chromosome and doesn't occur on the Y chromosome, because he didn't know that at the time. And so if you were just looking at this and we're assuming that this was an autosomal trait, you would assume that this was on a normal chromosome, this was on a normal chromosome, this was on a normal chromosome, and this was on a normal chromosome. And so this individual can produce one type of gamete, this individual can produce another type of gamete, and when you get this, you would get a heterozygote, and because this is a dominant allele, this would produce red-eyed flies. So assuming that you didn't know that this was linked to the X chromosome in a sex chromosome sense, you got exactly what Mendel would have predicted. A bunch of all red flies in the F1. But now, if we take these red-eyed flies that are presumably all heterozygous and cross them, let's say this is the female and this is the male, what would we expect to get? What gamete types can this individual create? A wild type and a mutant, right? What type can this individual create? A wild type and a mutant. If you put those into a Punnett square, we get homozygous wild type, a heterozygous that is going to show wild type phenotype. We get a heterozygous that shows wild type phenotype, and we get a homozygous recessive that shows white eye. So these guys are red-eyed, 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 and white-eyed. Phenotypic ratio of 3 to 1. 3 red eyes to 1 white eye. Yeah?
Are all recessive alleles non-coding genes?
No, not necessarily. We talked about this when we talked about loss of function. A lot of them are, and some of them are really obvious. When we were talking about Mendel's peas, flower color, the dominant trait is purple and the recessive trait is white. That's basically in one case it produces the pigment, in the other cases it doesn't, and that's due to a loss of function mutation. So a lot of recessive alleles are loss of function alleles, but not all of them. Well, no, that's also a loss of function allele. I was thinking of blood type. Blood type has multiple dominant alleles and one recessive, but the one recessive is actually a loss of function allele as well. So it's difficult for me to think of an example where it's not a loss of function, but I know that there are some that aren't. Maybe I can try to look up some of those and bring them back on Friday and let you know.
Yep.
Not necessarily, but in many cases, the majority of cases it is. So what clued Morgan into something might be weird going on in this? Because you got a 3 to 1 ratio. Three red flies to 1 white fly. What was different about the results of this cross? Every time he did this cross, it came out the same exact way. And it came out in a very peculiar way. What's the peculiarity? White eyes are always males. Females are always red. So what could explain this? What could explain this is that these male flies didn't have a recessive allele coupled with a dominant allele. They had a dominant allele coupled with essentially nothing, or if you want to, a different kind of chromosome. A chromosome that doesn't possess a counterpart to the dominant allele on the X chromosome. So if you replace this with Y, these genotypes become dominant allele with Y, recessive allele with the Y, and so half of the males will always be red-eyed and half the males will always be white, and the only place that you'll ever see white eyes if you have a red-eyed female mating with a red-eyed male that are the result of crossing the F1 cross from two parental pure breeding lines, you'll always see only white eyes in the male. Yeah.
So would that change the probability of a white-eyed since it's a 1 in 2 chance for a male, or would it still be one, like since it was 1 in 4 having white eyes, would it actually be like 1 in 8 since it's
Yeah,
one
so-
chance for males? To have
No, it's still a 1/4 chance because you have 4 possibilities and these are done. Yeah. But now the expectation is different. So you have a 100% probability of getting red-eyed females and you have a 50% chance of getting a red-eyed male. And so anytime that you see that these expectations are different by sex, that's generally a sign that you have some sex linkage going on. So,
So can you never have like homozygous recessive females?
oh, you can have homozygous recessive females.
Then why are they not white?
In order to have a homozygous recessive female, so answer this question yourself, what would have to be different about this cross? This is actually a good transition. Put your critical thinking cap on. What would we have to change about the parents here to get white-eyed females?
make it sex-linked for female.
Well, no, it's X-linked right now. So this is an X-linked trait. It's only found on the X chromosome. It's not Y-linked. It only gets expressed in the recessive situation here because there's no counteracting allele on the other chromosome because it's a Y chromosome. How could we alter this so that we get some white-eyed females?
Would the
Widely
the male parent have to be white-eyed?
The male parent would have to be white-eyed. Come up here and work the cross and show us that what you say is true. It's just a simple Mendelian cross of one trait. Wait, you didn't change things. I changed the
Oh.
genotype, but what did you not change? There you go. Change the gametes. So now you have 50% of your females are white-eyed. 50% of your females are white-eyed and 50% of your males are white-eyed, but you do get white-eyed females. But in order to get that, you had to have a white-eyed male. Okay? So you didn't need me to answer your question. You can answer your question yourself, which is good. Other questions? All right. And what's on the next slide?
What you just asked.
What I just asked. In order to get white-eyed females, you have to have a white-eyed male that you're mating to. Remember before, we were crossing two red-eyed individuals, a red-eyed male and a red-eyed female. To get white-eyed females to appear, you have to set up a situation where you can get the recessive allele from the red-eyed female coupled with the recessive allele of the white-eyed male, and that will produce white-eyed females down here exactly where you had it. Square. So when you ask that question, I'm like, oh, that's the next slide. We'll just do it on the board. All right. So what happens with this oftentimes is it sets up this form of inheritance where if you have a homozygous recessive in the female, you will see that phenotype crossing over and appearing in the offspring of the male. And the reason for this is if we keep this same Punnett square that we're working with, we now have homozygous recessive coupled with a red-eyed male. This individual can only produce one type of gamete. So we no longer have a 2 by 2 Punnett square, we will have a 2 by 1 Punnett square. The male gametes can go up here. The female can only produce the recessive gamete, and so it will produce heterozygous females and hemizygous recessive males. And so we have white-eyed females. They have to be white-eyed females because this is a recessive trait, and if there was a dominant allele in there, they wouldn't be white-eyed, they'd be red-eyed. And it produces white-eyed sons. And so this is another one of the kind of hallmarks of X linkage. When you have the recessive trait present in the female and the dominant trait present in the male, the dominant trait appears in female offspring, the recessive trait appears in male offspring. And so in this case, there's a 50-50 mixture of the phenotypes in the offspring, but all of the females are red and all of the males are white-eyed. So this is what your book refers to as criss-cross inheritance, is another one of the hallmarks of an X-linked trait. Not your typical 3-to-1 phenotypic ratio. As soon as you start seeing non-expected phenotypic ratios in data, you have to start thinking, oh, this is something other than just simple dominant recessive on autosomal genes. So So, if you have a 1 gene, 2 allele system, the alleles are strict dominant recessive, and they occur on autosomal chromosomes, you can expect to get a 1 to 2 to 1 genotypic ratio, and you can expect to get a 2 to 1 phenotypic ratio. If it's not those things, don't expect these things. And so anytime you see phenotypic ratios that don't match this pattern, that's a signal to you that something else is going on. In some cases, it may be X linkage. In some cases, it may be Y linkage. In some cases, it may be the alleles are dosage types of alleles, such that the more you have, the gene product changes the phenotype, et cetera, et cetera. So these are what we kind of think of as the Mendelian expected ratios. And other ratios, like a 50-50 ratio, would be an unexpected Mendelian ratio. Questions about X linkage? Now that I've done this, I feel like I should have altered this slide, this next slide. And I can't alter it because I'm already in it. What do you notice about this pedigree? Red is a hemophilic male. This is the royal family in England. Hemophilia is a recessive trait. How do you know it's recessive? No, that's how you know it's an X-linked trait. How do you know it's a recessive trait?
Because if it's a parent and its offspring, like, show that they may not have the phenotype.
Here are 2 unaffected parents, and what do they give rise to? They give rise to affected offspring. So if you have unaffected parents, they give rise to affected offspring, you know that, well, you know 2 things. You know that the affected trait is a recessive allele caused by a recessive allele, and it's harbored in what in the parents? Are these guys homozygous or are they heterozygous? They're heterozygous. They're both heterozygous because that's the only way that they can be unaffected by the trait, have it appear in the offspring. And so, first thing that you know when you look at this pedigree, just based on that top family, two unaffected individuals giving rise to at least one affected offspring, you know that the phenotype that is showing the affected offspring, that phenotype is caused by a recessive allele, so that's the first thing that you know. And then, as Chris pointed out, who's affected?
Dudes.
It's all dudes. There's not a single affected female up there. When you see this much representation of males exhibiting the recessive allele and no females exhibiting the affected allele, then that's the first signal that you look at and say, oh, this is an X-linked trait because it's being transmitted from mother to son.
Wait, so what does it mean to be a carrier versus just a human?
Okay, so is Queen Victoria a homozygous recessive?
I thought you said they were both heterozygous.
They're both heterozygous because she's unaffected. So what does it mean to be a carrier? I'm going to make you answer your own question again. Sorry, it's a rough day for you.
I don't know, that's what I'm asking.
Okay, what was the first cross that we did with these flies? We took a red-eyed female who was pure breeding, crossed it with a white-eyed male. What did we get? We got all red offspring. But when we took this red-eyed male and crossed it with this red-eyed female, what did we get? We got 3 offspring that were red. 2 of those offspring were female. 1 of those offspring was male. The other offspring was white-eyed and male. Why? What's different about this female as opposed to the previous female? She's heterozygous. So you get this allele combining with the Y chromosome here. That gives you white-eyed males. So note in the pedigree, does every male
So
have hemophilia?
So when they're, you can't have a heterozygous female that's not a carrier.
That's what it means to be a heterozygous female. You are a carrier. Yep. So you're carrying the recessive allele that when combined with a Y chromosome can make poor Leopold appear hemophiliac. Now, the X chromosome that you got going into Edward the 7th was the dominant allele of the 2 X chromosomes in the female, combined with the Y chromosome from Albert, which then made him not hemophiliac. So he had normal because you got the dominant allele from here, the Y made him male, and also made him normal. So you don't see all the males being affected. You only see some males being affected because they're in that 1/4 of the box of the Punnett square, because Queen Victoria was a carrier of the allele. You know she's a carrier because she's a normal wild-type female giving rise to mutant offspring. And when Alice was born, she got the X chromosome, which is hemophiliac carrying, and a normal X chromosome from her mother, which then made her a carrier. So she's now heterozygous as well. And when you're heterozygous, you have the possibility of passing off hemophilia to poor Rupert. Yeah.
So are all of the offspring males technically carriers?
No, they can't be carriers because males are X, Y. So for them to be normal, they have to have the dominant allele. Because this is what it means as a human to be male. So yeah, they don't carry anything. they carry the dominant allele and puny little Y. have hemophilia, this type of hemophilia, who do you have to blame? You have to blame your mother. Because your dad, all he's contributing to you is puny little y. You're getting either a normal, non-affected X chromosome from your mother, or you're getting an affected recessive chromosome from your mother. Nothing against mothers, nothing against Queen Victoria. So this is a classic famous case of X linkage in humans that is really easy because we have a nice pedigree of families. Okay, so then the question might be, well, how do you get this rare recessive allele appearing in the royal family to begin with? Anyone want to hazard a guess based on this pedigree? Inbreeding?
That's what was the Russians.
What do you mean inbreeding?
Oh.
First cousins. One of the consequences of repeated instances of inbreeding is you get the fixation of genotypes, and you also get the accrual of recessive alleles typically. So, we won't think about it too much. It's what being royal makes you. All right, we have 3 minutes. Let's just knock it off. We'll talk about non-disjunction next time because it takes a couple slides to get through, and we're almost out of time. Any questions? Do you learn more when I dance and sing?
Yeah.
All right, I'll see you guys on Friday. Yeah. In the pedigree? Oh, sorry. The mom is heterozygous. The dad, he's heterozygous. I introduced this term. Hemizygous is like half, specifically half zygous. And what it means is that you have one chromosome that is full versus empty. So you might have the gene here, but the Y chromosome is not full, so that section of the chromosome carries that gene in there. So whatever is on the X chromosome is just not there.
Sorry.
It's actually, it's found on the X chromosome of the sex chromosome. So autosomal chromosomes are all the other. There wasn't rush. So if he wasn't, so all of these other chromosomes, all of these are autosomes. These are sex chromosomes because the chromosomes differ, and that difference determines the sex. So we have 20 pairs of chromosomes, 22 of them are autosomal, and 1 pair of chromosomes.
What's what we talked about in?
So when I said heterozygous, I should have said
I just don't remember
non-heterozygous.
exactly what it was.
I know that shape is
All males are not
X.
homozygous.
Not in all categories, it's all humans.
Yeah, he did. I think only some of them developed.
Was
I'm sorry.
if you
Are you actually
X?
XXY?
Only if she's a carrier. Only if she's a carrier.
So I'm like, if there's a Y,
Yeah, because we don't ever.
How would it be
so
for
let's say
men?
we don't
Yeah.
have the recessive allele
Like
from your mother.
Klinefelters?
There's no possibility of
It's
an
Klinefelters?
equivalent dominant
It's all-
element from your mother because that chromosome doesn't even have.
it's
that.
not familiar. One of them's for like.
Yeah, Caleb.
Slide will be able to check the times.
Oh, were they not up?
Did you post it? Cause I had to check in like a day, so.
I thought I did, but I'll check again. Yeah.
Okay, thank you. And then these will be up after we finish this.
These will be up probably later on today.
Okay, sounds good.
Yes, ma'am.
We were talking, some of the life we learned about if you're female but you have, is it possible that you have a Y chromosome if you're XXY? Is there some kind of